coordinator for AICRP, honeybees and pollinators. Previous to this assignment, he was principal scientist in Division of Entomology, IRI New Delhi, looking after biological control section, and now he is looking after the AICRP, honeybee and pollinators. So I welcome you, sir, and he will be talking on trends in beekeeping and pollinator research in India. So welcome, sir. चेयर पाहिजे
चेयर मदत आहेत का ते
नाही
$12.15 ना ठीक
one list I have circulated. Just you check your spelling, name spelling. If there is any mistake, you can correct it. And those who have registered but name is not there in that list, they have to write their name in block letters. Okay.
So good morning, students. Hope you all are fine and your stay is also okay. So as told by Dr. Suresh, I am looking after the All India Coordinated Research Project on honeybees and pollinators. And you know, honeybees, they are such a important to us, as not that they give us honey, pollen, nectar, then we call royal jelly, then bee venom, but they are also responsible for the ecosystem services in the form of pollination. You see, with respect to our AICRP, it was actually in 1976 there was National Commission on Agriculture. It was in 1981 it was established at CBRTI Pune, and in 1987 it was shifted to Hisar. Then in 2007 the title was changed. They have added pollinators also. So you know that not just it was AICRP on honeybees, but they have added other pollinators also into the realm of this project. And the centers went on increasing day by day, and so it was there in Hisar from 1987 to 2014. Then in 2014 it came to IRI New Delhi. So we have PC unit here only in Division of Entomology, and presently we are having 25 centers. Out of these 18 centers they are regular centers, and 7 centers they are voluntary centers. You can see we have the AICRP centers in different parts of the country. So except Madhya Pradesh, we are having centers in each of the state now. And you know, there whenever there is project, there will be objectives and there will be aim. So we know until now we were concentrating lot of our efforts on the honeybee research. But you know, now pollinators, they pollinate around 85% of the agri-horticulture crop. So Government of India wanted to conserve them. Then what are their economic value with respect to pollination? Many times, you know, we see insects on the plant and we see they are pollinators, but many times they are visitors. Okay, but you need to study in detail what is the pollen-pistil interaction, whether the pollens are viable or not, and what is the value they are contributing towards the pollination. So there are so many activities we are doing. Then what are their roles in self-pollinated as well as in cross-pollinated plant? And pollinator decline, you know, now with the habitat destruction, habitat change, climate change, we are losing so many species day by day. They are going on extinction. And you know, whenever we talk of insects, the taxonomy's role is very, very important, you know, because we are having so many non-apis bees and we do not have enough taxonomies to address this issue. Then we are also working on its conservation. So with respect to history, you know, in India in 1884, the father Newton, he has designed the smaller hives for the Apis cerana that time. And with that, in India, the apiculture industry flourished. So in 1937, the R.N. Mattu, he founded the All India Beekeepers Association, and they have taken forward so many research and extension activities. So In 1954, Government of India, they have established the KVIB, that is Khadi and Village Industry Board. It is part of Ministry of Small Industries, Small, Medium and other industries. And in 1962, the CBITI was established at Pune. Then we have introduced this species from the Italy in 1962. It was imported in Nagarota Bhagwan. Actually, it is in Himachal Pradesh, but that time it was a part of Punjab. And there is lot of scope with respect to honeybees and pollinators. We have crop area around 160 million hectares. Then out of that, crops which need pollination, so area which on which the crop need pollination is around 55 million hectares. And beside that, we have a lot of forest area, pasture land, wasteland, and lot of cultivated and uncultivated lands are there. And India is what you call leading mega diversity centers in the world. And we have 4 important honeybee species, plus we have 20 stingless bee species, and we have around 700 species of bee flora. So, and with the introduction of Apis bee fellifera, we are having lot of scope. And the next 20-30 years, there will be a lot of research on honeybees and pollinators in the country. And with respect to the honey production, you see, few years back it was around around 70,000 metric ton, but now we are producing around, this is 2021 22 data, but 23-24 we have produced around 180 lakhs metric tons of honey, and we are exporting around 50% of it, around 80,000 metric tons we are exporting to the other countries, especially to the United States of America. And you have studied many times, we are having Indian honeybee, Apis serena, Apis flora, little bee, mellifera, dorsata, and we are having around 20 species of stingless bees. And there are so many strains of Apis mellifera. German black bee, Apis mellifera, then Italian honeybee, Apis mellifera ligustica. So we have introduced this species in 1962. And America, a lot of research is being done on carniolan honeybee, that is Apis mellifera carnica. And there are so many species. This African honeybees also there, Apis mellifera scutella. So presently, 95% of Indian honeybees, colonies which we are having in India, they belong to the Apis mellifera ligustica. And when we talk about honeybees, we normally concentrate on honey. But this is normal. This is old data actually. That time, one colony used to give 50 kg of honey in 1 year, 4 kg of pollen, bee wax 800 gram, royal jelly 823 gram. Propolis 300 gram, and bee venom. Apitherapy, it will be big industry within 10 years, 20 years across the country. But due to climate change, loss of bee pasturage, bee flora, we are not getting 50 kg nowadays. We are getting around 25 to 30 kg. And rates are also quite high. Royal jelly, actually, we are having very good technology in India, where we have identified some molecular markers also, which are responsible for higher royal jelly production. But when honeybee want to produce 1 kg bee wax, they need to drink 8 kg of honey. So there is a lot of cost involved with respect to bee wax production. And you see the current prices. Honey you can get around 100 rupees. You need to pay 4,000 rupees per kg to get good honey. 4,000 I am talking about the stingless bee honey. So you get around 5,000 rupees also. So bee wax, and just see the royal jelly rate, around 32,000 per kg. Taiwan and China, they are leading. They are producing 70,000 metric tons of royal jelly. And Japan is the main importer. They are using in cosmetics, they are to treat newborn babies, for the treatment of cancer, then sterility problem. So royal jelly will be one of the major medicine in coming years. And apart from this, you see, one beehive, we can get you 5,000 to 7,000 rupees with live bees. One queen bee costs around 500 to 1,000 rupees in India. Then pollination services, if you keep colonies for 20 days or 1 month, so you need to pay 1,000 to 3,000 rupees per colony for doing pollination. And role of pollinators, I already told you that 87% crops being pollinated by the pollinators. And the area, so crops which need pollination, their area increased by 300% during last 50 years. 80% of the pollination is performed by the insects, and there are so many crops. If there is no pollination, 90% of the crop yield can be decreased. And this is one of the statistics. You can see, as I told you, there are around 20,000 bees in the world, and we are having around 800 apoids in India, and they contribute around US dollar 577 billion towards the animals as a whole for the pollination, and insects, out of which the insect contribute around 161 billion US dollar through pollination. And there are so many solitary bees like alkali bee, blueberry, carpenter bees, so many species which also contribute. So you see, wild bees, they contribute around US dollar 3,251 per hectare through pollination. And with respect to success stories with respect to pollination, so we have carried out these studies in apple, and you know, apple is totally cross-pollinated crop, and you know, if you eat the apple, it is being pollinated through the pollens from other varieties. So crab apple, you know, it is one of the pollinizers, and there are so many types of pollinizers. So if pollen comes from other varieties, then only apple will be pollinated, it will fertilize, otherwise you will not get apple. And you know, we have done these studies, bee colony with pollen dispenser and bee colonies without pollen dispenser. And you can see here 25 meters, 50 meters, so distance of this bee colony from the orchard. So if you are keeping the bee pollenizer within 25 meters of the orchard, you see the apple fruit set, it is around 70%, and if you are going on increasing the distance, the fruit set percent goes down, and you can see the yield around 60 tons per hectare you can get with bees with good pollinizers, and without pollinizer you can see you are not getting even 10 tons. So there is a very good scope for using honeybees and other pollinators in apple pollination. The same case with respect to cherry, you can see. So when you have colony within 25 meters of the orchard, the fruit set goes up, and as you are going on increasing the distance from the orchard, the fruit set goes down, and you see the most percent fruit set also. With bee pollination, there is very good. And you know, we have introduced BT cotton in 2001, and there was a report that BT pollen might affect the health of the honeybees. So that we'll see in other slide, but there is no adverse effect on the pollen hoarding or nectar hoarding with respect to honeybees. But cotton is very good source of nectar, and it gives the nectar during September to November. Normally we don't have flora which gives nectar during this time. Honey flow season starts from November to March. So this is a very good time, and you see the seed yield, seed of the cotton with respect to BT, non-BT cotton, and in open pollination there is always good performance, but bee pollinated crop is also giving very good seed yield compared to the pollination exclusion experiment. And not just honeybees, we are using bumblebees. Bumblebees, you know, they are very good pollinators, and we are having around 60 species in India, 10 species in Himachal, 10 in Jammu, 10 around in Kashmir. And we are devoting a lot of research on Bombus hemorrhoidalis with respect to the pollination. So here you can see in next slides, with bumblebee you see fruit weight can go up to 413 gram compared to the other. Open pollinations will always give you good result because you get diverse source of pollen from other sources. Same case with respect to number of seeds per fruit, weight of 1000 seed also very good with respect to bumblebee pollinated crop. So pollination is not just to get the fruit yield. So many times you know, there is seed industries, they are using onion seed production. Honeybees, if they are there, you get 3 times more seed yield. So they are playing very good role in seed production also. So you know, tomato, you know, it is a cross pollinated crop, and it needs a buzz pollination. Buzz pollination means they are having poricidal pollen, and unless some pollinator they shake the plant, pollen will not come out. And bumblebee, they are very good buzz pollinator, and you can see the yield. You get mean fruit yield around 6 kg here per meter square, and you can see your seed yield, fruit yield goes double, around 12.7 kilogram per meter square. And recently there is very lot of price hike with respect to the tomato and onion. And the Israel being very small nation, they are exporting their what you call 100% tomato to the Europe. And if we are having very good pollinators like bumblebee in our Indian scenario, we can also produce what you call we can also produce more quantity of tomato. So not just bumblebees, carpenter bees also we are targeting, and we have already established the nesting structure. You can see bamboo having a hole of one what you call 10 millimeter radius, and bumblebees, if you offer them nesting structure, they can make their nest and they can do pollination. And in rich gold, cucurbit, they are normally monoecious crops. So male and female flowers will be on same plant, but their location will be different. So we can observe 250% increase in the yield with respect to the carpenter bee pollination. So these are the studies which we have carried out last year with respect to what is the role of different pollinators. And not just carpenter bees, bumblebees, blue-banded bees. They are very good pollinator in solanaceous crop. So we have carried out this experiment using 1 bee visit, 2 bee visit, 3 bee visit, 4 bee. As we went on increasing the bee visits, though the length and weight of the flower, this chili fruit increased. Then seasonal management with respect to Aphid mellifera, I already told you about the performance of Apis mellifera on BT and non-BT cotton. And there is brood survival, there is no big difference when you use the pollen and nectar source from BT or non-BT crop. And we found that the 3 feedings of sugar, that is around 500 gram in equal quantity of water at 10 days interval, it can give you very good colony strength, brood rearing and honey storage. And winter packing, in North India we get very severe winter. So we have used the paddy straw wrapped in polythene to increase their hive temperature, so that when the hive temperature will be maintained, then brood area also can be increased. And normally after honey extraction, you use the old combs for colony development. So 1 and 2 year old or fresh combs will be better compared to the 3 and 4 year old combs. So one of our center in Assam, it is you get around more than 1000 mm of rainfall in northeast, and when it rains, it might affect the colony health. There will be a lot of they don't venture outside for foraging, and the using this bamboo hat, which they called as that japi, so the temperature also can be maintained, and the brood area, everything, honey, the other parameters also will be good. And Apis serena, the Indian honeybees, it is prone to absconding, and we have carried out our experiment with respect to how to minimize it. So how we can reduce the absconding less than 12%. So we have all the technology with how to do colony reunion. So suppose the strength of the colony goes down, then you have to merge them. Otherwise, if the colony become weak, they will migrate. So that feeding, how you should do the colony division, then you follow these things, then there will be new comb constructions within few days. As you know, you are now undergoing training on genomics. You see now we are identifying the markers which are responsible for royal jelly production. As I told you, Taiwan and China, they are the big producer of royal jelly. We are having all the technology, but in India there is no demand, and this will be the new medicine in coming years. So we are doing research to find out the colonies we are having, which are having these 2 genes, 2 alleles, MRJP3 and MRJP8. The colonies which are having these alleles, so you can see the production of royal jelly can go up to 350%. It will be, and likewise the colonies having high hygienic behavior and the colonies which are having less defensive behavior. When we extract lot of bee venom from the colony, they became too defensive. It will be very difficult to extract the honey and to do the day-to-day activities. So we are finding out the genes which are responsible for this characters like higher royal jelly production, as well as they should have high behavior. If they are having very good hygienic behavior, means the colony can fight nicely against the diseases and other biotic stresses, and they should have less defensive behavior, so that they do not bite. And normally the colonies which are collected from hill zones, they show very good performance compared to the colonies which are collected from plains. And earlier the Indian bee was named as Apis serena indica, but now we are two distinct species now: Apis indica from the plains and from hill Apis serena. So 7 frame hives, they always perform better compared to the 6 to 8 frames hives. And now stingless bees research is increasing day by day. As the stingless bee honey, it is having a lot of medicinal value, and as we get 5,000 per colony form for Apis mellifera. So we can get around 10,000 per colony for stingless bees. But problem is maintaining the colony. So if the colony is queenless, so the colony multiplication rate will go down. So we have come up with the studies that we can use the pupa of queen cells, and moment the colony multiplication start getting fast. So as our Solon Center is working on Bombus hemorrhoidalis, that is our Indian bumblebee, and they were having problem. They were using wooden domiciles for his multiplication. So both of our scientists, they went to South Korea, they had training, and they brought the plastic starter boxes, and they are giving very good results. So we may have bumblebee technology for pollination in protected cultivation. And with respect to the nesting structures for the Xylocopa carpenter bee and mega child, that is the leaf cutter bees, they are very good pollinators in pigeon pea. So we found that the bamboo nodes with 1 centimeter diameter, they are giving very good result with respect to the nesting structure for Xylocopa. And mega child bees can be reared, or they can be offered the nesting structure in the form of sarkanda. Sarkanda is one of the weed with which is related to bamboo family. And as I told you about the yellow banded wasp, Haplonomia westwoodi. So if we want to employ them for pollination, we need to give them the nesting structures. So we have given them the types of nesting structure, like red soil with sand, they are having different combination, and red soil with FIM. At the same time, this nesting material was offered to them in polythene bags having different dimensions. So we found that the polythene bag having 20 of size 25 by 40 centimeter, and along with red soil with FIM, it was found to be the preferred substrate. So you give them, they make the nest. You can see the nest, and we can carry out the pollination in protected cultivation. And within last few years, we are getting a serious attack of chalkboot disease in Tamil Nadu, as well as thigh sac brood virus in Odisha. So we are constantly working, and this is the new pest which came into India last year. This is the invasive pest. It was not there in India until now. So we have reported it for the first time from the India. And the good thing is that actually they pupate in the soil. So I think within next 4 to 5 years or 10 years, this pest will not be a problem. We can manage it nicely, because grub has to fall in the soil, they pupate it there. So one of my student who is undergoing training, Loganathan, you are there today? Yeah. So he is working on his total management aspect. So he went to northeast, he has done the survey, and it is not just affecting Apis mellifera, it is affecting Apis serena indica also. So with respect to the disease management in apiculture, European fall boot, it is a very dangerous disease and very dreadful, and we cannot use the antibiotics. Antibiotics, once they go as a residues in honey, so we cannot export the honey. So we are finding out what are the non-chemical options available with us. So we have neem oil, rosemary oil, and green bee eater bird. It is a big threat for migratory beekeepers. So when we migrate colony from one place to other place, so it is actually the Apis mellifera is the actually very susceptible to this green bee eater. And our Indian honeybees, actually it is not that susceptible to this green bee eater. So we are working on how to get rid of this green bee eater bird. And we also tried some probiotic diets in honeybees, so that how to restore their cell integrity in the midgut. So pesticide toxicity, it will be always problem. If you develop any pesticidal molecule for any insect pest, it definitely it will be affecting honeybees. So we need to spray early in the morning, late in the evening, or you should not spray when there is a bloom period. And we have carried out the what could be the impact of elevated CO2 on Apis cerana. And there were 3 combinations: 380 ppm CO2, 650 ppm CO2, 750 ppm CO2 with increased temperature. And when compared to the field visit of the honeybees, when they visit the crops sown in the open top chambers having high level of CO2 and high temperature, foraging frequency goes down, and their pollen load per trip also goes down in elevated temperature, and the direct impact on the yield of honey also, mustard yield also, you can see there is a less yield in the plot having elevated temperature and elevated CO2. So if there is climate change, it is going to affect the pollinators and honeybees. So we have also standardized the technology for the extraction of bee venom. So you can use install the bee venom collector for 60 minutes only for weekly interval. If you use it for more, then colony will become defensive and they will start biting. So it will be very difficult to work in the apiary. So you can at least have, in 60 minutes you can get around 15 milligram, 14.69 milligram of venom. And if you are having colony with 16 bee frame, then you can get around 20 milligram. And we also found out that when instead of keeping the this venom collector near the entrance, you can keep it on the top bars inside the colony. You can get around 18% more yield of the bee venom. And we have compared the imported and indigenous venom collector, and we found out that indigenous they cost 25% less and also produce more venom. And we have also tried the effect of artificial diet on the bee venom collection. So you see, when you give them natural pollen with sugar solution, you can get around 18.47 milligram of bee venom. But suppose you are not having natural pollen, during dirt period you will not have. So one of our center, that Louisiana center, they have developed their pollen substitute. So still we can get around 14 milligram of bee venom. Royal jelly production, I told you, we are already having, we have standardized the technology for production of royal jelly. We need to have molten wax, then we need to prepare queen cell cups. So if you pour this molten bee wax in this queen cell cup, you can get the queen cups. So you can put around 20 cups on one horizontal bar. So we are having 3 here, so 60 queen cell cup you can have on one bee frame. And you need to transfer the grub of the honeybee, which is less than 24-year-old, in this queen cups. And once you have transferred the bee grub on this queen cell cup, you transfer this colony in the colony having the 15 to 20 bee frame strength. Then the nurse bee, they attend to it, they secrete the royal jelly from their mandibular gland, and after 72 hours you can remove this queen frame from the main colony, and you can cut the queen cell cup at the level of the royal jelly, and you can extract the royal jelly. So the beekeepers which are getting lot of revenue through honey, pollen and pollination, they are going to earn lots of rupees with the production of royal jelly in the future. And as you know, we normally talk that stingless bee honey is having a lot of medicinal property. What are those? It is having more content of the calcium, more content of the potash, more content of the sodium compared to the Apis mellifera honey. And you can see the total phenol and total flavonoids, which are a very good antioxidant properties, they are also on higher side in stingless bee honey. That's why consuming stingless bee honey is very, very good. We are also working on how to what should be the technology for the pollen collection using both Apis indica and Apis cerana. sorry, Apis mellifera. So you can see the study was carried out in sunflower mustard and Niger. So if you collect once a week, so you can get around this quantity of pollen, and pollen also get you around 15 to 20,000 per kg. It is very good source of protein for vegan people. So same studies with Apis mellifera we have done. So we are already having the technology. If some farmers they want to they produce pollen only. So we are already having technology in which one they have to do and in which one they should not do it. Conservation of Apis dorsata. Normally you see we consider dorsata as very ferocious bee. Normally you see them on these big buildings, rocks, hostels, big buildings normally, and you know they are the main honey gatherers in the Sundarbans forest. You know we are having 6 species of mangroves. Mangroves you must have seen if you might have visited Goa, and you see the honey flow season is normally from March to May. Normally this time there will be very harsh climate in this part of the country. So, and you know we used to lose around 25 to 30,000 colonies per year. You know these honey hunters, they call them Maule in actually Bengali, this must be Bengali term. They just kill them, they burn the colony and they collect the honey. And the colony, the quality of honey also will not be that good. There will be dead honeybees and there will be a lot of wax available. So what we have done, we have started giving them the training. You see, we have counseled so many traditional honey hunters. We have given them scientific knowledge about the honey collection, honey extraction, and we told them what is the importance of pollinators. So we should not kill them. So last year we could save around 874 dorsata colonies. So we can save more than that. And you know normally when they go for honey collections, there will be fictin man and animal conflict. You know that Bengal Royal Bengal tiger, they attack the honey hunters many times. So this is what we are doing there. We also established a very good model bee garden at GB Pantanagar. You can see the flora there. So this is a new plant, solidago. We are trying, where it is very good source of nectar and nectar and pollen. And you know, when you do any research, we need to empower the entrepreneurs. So we have formed the 3 farmers producers organizations. You know, one in Satara, one in Nashik, and one in Gadchiroli. So they are the people who can do lot of work with respect to value chain in honeybees and pollinators. And some of the centers, they have brought out so many products so that they can get extra revenues. These are some of, you see, we are having honey drink, honey jam, bee wax lip balm. It is, we have given the cost, what is the benefits cost ratio also. And this is the one of the FPO which to whom we are guiding, and these are the products which are being sold in supermarkets. So thank you very much for patience hearing. If you have any question, you can ask it. Light love, Suresh. So first let me know who is eating honey daily. आज किसने खाया honey? Who have consumed today? Okay, before coming here, anybody consumed honey? When you had it last time?
Last 1 month I taken, sir. Last 1 month.
1 month back.
Sir, in past 1 month I am taking regularly.
So who told you to take? Just. So who is regularly eating honey? Don't fear. I am one belong to you only. Daily. So how you consume it? No, how you are consuming it? Daily. So what you do? First you boil the water. So what will the temperature of water? Can you put your finger in that water? Then you add honey. That is not good for health. It is poison. 50 degrees Celsius. So the temperature of water should not be more than 45 to 50 degrees Celsius. That is the normal temperature honey can sustain. Otherwise, you are eating poison only. Don't drink that one. Okay? You should be able to dip your finger. Okay? Lukewarm water you talk normally. Hai na? So that's good you are taking. It's very good. Anybody else who is consuming honey daily? So what do you think? How much we are taking as an Indian, one person daily? How much we are eating? Anybody can tell. Just guess. Guess also. You can guess also. That is why we are not producing that much royal jelly, that much honey compared to China, because they are having demand. In India, there is no demand. We are using honey only for religious purpose, for making panchamrut. है ना? So what could be the per capita consumption in India? You tell me per day what you must be eating. बताइए. She is telling something. You only. Huh? Per day. So yearly how much? If she is telling 5 grams per day. So year how much it will be? 30 gram. 300 gram actually. है ना? 300 gram in 1 month. है ना? 300 ml. So बात ही 36. So how much she is telling? Maybe 3 kg. 3 kg only ना? 1.5 kg. 1.5 kg per year. So anybody? So that is, she is telling 1.5 kg per year. Okay. Anybody who is agreeing to her or disagreeing to her? आप बताइए madam from Kashmir. You are not eating honey? It's not good. You belong to Kashmir and it's very cold. Honey is very good source of heat. You know, if you consume it, it gives you a lot of comfort against the cool temperature. So how much one person is eating in India? So she told at least we should have 2-3 more answers. भाई वीरेश बताओ आप. वीरेश कहाँ है? वीरेश कहाँ है? वीरेश is there? हाँ वीरेश बोलो. 2 kg. India. Okay. So you are eating 2 kg every year. Then average you are telling. Okay. Anybody? आप बताइए. 100 gram per year. Okay. So per day how much?
Per day, like it will be like on per capita, sir, it will be less than like 1 gram.
So whether you are agreeing to him, we are eating only 35 gram per year, one person, one Indian, है ना, per year, and Europeans they are eating around 3 to 5 kg per year. So you see, so start consuming honey at least 5 gram, 1 teaspoon, like she is taking. So you can have at least 1 or 1.5 kg per year, and there is no demand. That's why there is no actually how much farmer must be getting in the. Suppose anybody is having apiary, you are from Jammu, hai na?
Yes.
So what is the per kg rate?
So it's 500 kg.
That is you are selling.
Yeah.
How much beekeeper is getting when he is selling to companies?
200.
200. If they get 200, they will be very, very rich. So tell me now how much they will be getting. They are getting around 80 rupees per kg.
No, sir, but we have talked to some
No,
beekeepers.
I am-
They told 200.
You are talking. I am telling you correct data. I am part of that scheme. ठीक है? 80 rupees only, 90 rupees only they are getting. Even though if you give them 150 rupees, they are very, very happy. That's why there is lot of adulteration with respect to honey sale in India. है ना? They add corn syrup, they add rice syrup, they add oligofructosaccharide, OFS, you know? OFS, anybody from post-harvest technology? We import it from China as a sweeteners for biscuits or chocolate making, and they are adding it in honey. It is very bad for liver and kidney. So actually, what is the test if honey is not adulterated? Anybody knows? If you want to check that honey is adulterated or not, any test? You can tell you are from that. You are doing research on honeybees only, I think.
Yes.
Yeah. So anybody is doing PhD research on honeybees? You are from which campus?
Cost.
MPUT Udaipur.
क्या कर रहे हो वहाँ पे?
Diversity and ecology of insect pollination.
That's good. Who is your guide?
Dr. S. Ramesh Babu sir and R. Swaminathan.
2 guides.
Yes, sir.
It's very good. Very unique combination. Ramesh Babu was my classmate when we had PhD here. So anybody, if is having any question, he can ask. Actually, honey, when you purchase the honey, it should settle at the bottom directly. ठीक है, it should be settled there and then it can start dissolving. That is only unique test. There are so many tests otherwise, but you cannot do, है ना, we cannot do regularly. So that is the one test you can follow, and try to purchase it from beekeepers if you know them in market. And the crystallization, you know, normally you see that the crystallized honey is not good, but pure honey only crystallizes, and especially mustard honey. So we get mustard honey in the North India, and it crystallizes, and 90% of the Indian honey it crystallizes, except bear honey and lychee honey in some extent. Okay then, thank you, all the best.
Thank you, sir, enlightening us about status of honeybee and other pollinators and its research in India. And some of few slides also you saw that how these molecular tools can be used in enhancing the production and other products of the honeybees. So thank you, sir. From sir. So on behalf of organizers and on my behalf, I wish to give a token of love to Sachin sir. So I request Subramaniam sir to hand over a small gift to him. Sir, Sachin sir. Thank you, sir.
Thank you, Suresh.
Thank you, sir. So now we will move to our next lecture, and already our faculty has come here. So next lecture will be by Dr. Sandhya Sharma, senior scientist from NIPB, New Delhi. So she will be talking on genomics, genomic sequencing principles and practices.
Welcome Dr. Sandhya Sharma. She is the senior scientist at the, please come, National Institute of Plant Biotechnology. She will be delivering on the advances in next generation sequencing and its application. She is a master in especially the NGS techniques in teaching students as well as using them for the plant biotechnological research. So we have a very good speaker to elaborate on the NGS techniques as well as the whole paradigm of the next generation sequencing. Now the floor is yours, Dr. Sandhya Sharma. Thank you.
Thank you so much, sir.
It is there. The lecture is also on online.
Okay. Yeah. First of all, thank you so much, sir, for providing me the chance to interact with your mind. Very good afternoon to all and all. Sorry for being late. So I have assigned a topic, genome sequencing, practices and application. So I'll be covering the basics. It is always good to start with basics, so we'll be dealing with the basics of sequencing, then we'll move to the next generation advances in next generation sequencing platforms, how one platform is different from others, what are the advantages and disadvantages of techniques, and then we'll have some idea like how to prepare a library if you are planning for RNA sequencing or transcriptome analysis. Okay, so yeah. So I would like to begin with a very trendy terms which is very catchy nowadays, omics. So Can anybody will explain what is omics? What is omics? What do you understand by omics? Yes, please.
Wide study of genomics, the wide study of genome.
Yeah. Anybody would like to add something? So, omics is nothing but the, you can say, study of totality of something. Sir, can I move here? Or, I mean, if it is online, then I have to stick here, no? So, it's a study of totality of something. For example, if I say genomics. So, what is genomics? Study of entire genome. What is genome? Please, one by one, you can answer. Okay. Anybody else?
Total amount of gene in particular organism.
Very good. So, in genomics, we study the structural and functional aspects of genome. Transcriptomics, where we study the expression profiling, transcriptional regulations. Proteomics, it not only deals with the identification and quantification, but also post-translational modifications of protein can be studied here. Recently, this metabolomics and omics, these new omics have been introduced. So, metabolomics, as the name indicates, so it is something related with the metabolites or the study of metabolome. Okay, so here again, this metabolomics again on the basis of your requirement, it can be categorized like targeted metabolomics and non-targeted metabolomics. So, what is targeted? For example, you are given a oil or any things, food. You want to study or explore the lipid profile of that particular compound oil. Okay. So for that, you will go for the lipidomics or the targeted metabolomics. And in non-targeted, where you will be exploring the all sort of metabolites present. So it's basically the compositions, okay, of all the metabolites. In ionomics, what we do? We study the ions profiling. Why ions are important? I mean, why to study ion profiling? There are many enzymes which requires ions as a cofactor, so they are the integral part of metabolic processes also. Okay, and in phenomics, what we do? We evaluate the morphological, biochemical, and physical trait. So basically, these are the major omics in biological sciences, right? Now coming to the sequencing and why sequencing is important. So what is sequencing? Genetic code. इधर से
Conversion of variety signals into nucleotide sequence.
you can be slower.
Conversion of variety signals into nucleotide sequences.
Okay. Anybody from this row? What is sequencing? I think you have attended my class, no? Then you should answer.
Sequencing is just analyzing the arrangements of the nucleotides in a DNA.
Very good. So it's a process. It's a process of determining the order. Order of what? Nucleotide or bases if we are talking about the DNA sequencing, and order of amino acid in case of protein sequencing. So determining the order of nucleotide, how they are arranged one after another, ATGC, arrangement of ATGC is sequencing. Okay, and by sequencing you can have an idea about the coding sequences. Okay, what all are the part which is non-coding, what are the regulatory sequences, regulatory elements. Okay, and then if you want to study for example, if you identify a gene related to some particular abiotic stress or biotic stress, or which is nutritionally important gene. If I say you are working on any crop or any plant, so you want to if you want to do the salt tolerant or salt sensitive things, so how will you identify the genes which are responsible for salinity tolerance? if you are working on the trait salinity, salt tolerance in any crop. So what will be your approach? How will you do? There may be many genotypes, many varieties. So how we will start? Isolation of? Okay. Then?
Isolation of DNA first, then we have to identify which.
See, for example, if you are having first, you have to look at your collection. For example, if you are having 100, 200 or 300 different lines for your crop, okay, then first you have to do the screening on the basis of morphological, physiological, various biochemical parameters are there, okay. Then you will get what? One tolerant plant and one sensitive plant. Then you will go for the comparative transcriptome analysis, where you will get the whole total number of transcript expressed in the tolerant plant and then sensitive plant, and then you have to do the downstream analysis, and you can catalog what are the genes which are only expressing in the tolerant plant and the genes which are expressing in sensitive plants, what are the commonly expressed genes, and what is the expression fold, the level of if they are expressing significantly in tolerant plant. So like you can catalog to your genes, okay. So you can have idea about the genes responsible for any kind of trait, okay. And not only gene, transcription factor, and then the associated pathway you can identify. This is the slide showing general genomic comparison among organisms. So these are the organisms. This is the genome size and the estimated gene. If you look at the Arabidopsis thaliana, the genome size is 100 million and the estimated gene is 25,000. In fruit fly, genome size is more. It is 137 million, while the estimated genes are just half. What does it mean? Why I am showing this slide?
There is no relation between number of base pairs.
Very good. So it means there is no correlation between the size of genomes and then the complexity of the genome. Okay. So what makes genome more complex? What are the factors responsible for? There may be more repetitive sequences are there, number of intron, maybe there, pseudogenes are there, repetitive elements are there. Okay. So so this is the chronology of DNA sequencing. If you see this, it was discovered in 1870, and then the double helical structure model was given by Watson and Crick in 1953, and the first tRNA sequencing was performed by Robert Hooley in 1965, and the most common, you can say, and the popular methods of sequencing were evolved in 1977 by Sanger and Maxim Gilbert, and then after some evolution in technology, we got partial automation, then fully automated sequences came in picture, and then we got the complete sequence of S. cerevisiae in 1997. Okay, so and this is how the sequencing techniques were evolved. If you see, it started way back in 1977, and now we have number of sequencers in the market. So after the Sanger sequencing, the first commercial sequencer, that is Roche 454, you must be knowing this. It was introduced in 2005. Then Illumina came in 2007, and you can see here different variants of Illumina technology as well as Oxford nanopore sequencers. They came in between 2010 to 15. Okay, so coming to the sequencing method. So though there are various platforms, okay, number of sequencing techniques are there. So we can, for our understanding, we can categorize those sequencing platform as a basic and advanced sequencing. Okay. So basic will be talking about Maxim Gilbert and Sanger sequencing, and others will considered as a advanced type. Okay. So first we'll start with the Maxim Gilbert sequencing. What is the principle of Maxim Gilbert sequencing? Anybody? Have you heard about Maxim Gilbert sequencing? No? किसी ने नहीं सुना? हाँ. So this is the basis of sequencing. कितने लोगों ने Maxim नहीं सुना है? Can you raise your hand, please? Okay, I'll tell you. So in sequencing, what we are doing, we are just identifying the arrangement of ATGC. Okay, how these ATGC they are arranged. Okay, so the idea of Maxim Gilbert sequencing was to use of some chemical reagents. Okay, so once and those chemical reagents will be specific for the basis. Okay, for example, if I am adding dimethyl sulfate followed by piperidine, so if I are given a stretch of DNA like maybe 20 base pair, whatever it is. Okay, so and if I am adding dimethyl sulfate followed by piperidine, what it will do, it will cut at the G site, guanine. Wherever guanine will be there in the sequence, it will cut at G. Okay, so likewise we have specific chemicals for specific bases. Okay, this method is also known as chain termination method. Sorry, I'm so sorry, this is chemical degradation method, because we are using some chemical to cut the sequences, and this is the whole procedure how we perform. So we start with the double strand DNA, we prepared single strand, and then what we do, we add 5' phosphate labeling. So this is the preparatory phase. So what we do here, we prepare 4 set of reaction. Okay, 4 set of reaction means 4 tube, G reaction, A plus G reaction, T plus C reaction, and C reaction. Okay, and all the things required for the this will be added here. And here the chemical specific for guanine will be added in this tube. Okay, why I am telling this A plus G reaction? Why not only A? Because the chemical which we are using for eluting G, sometimes it also cut at A, because they are the purine. Okay, likewise we have a T plus C reaction and C reaction. So जहाँ जहाँ पे आपको chemical degradation, जहाँ पे आपको specific होगा it will cut. Okay, and you will get the different fragments like this. Okay, then what we'll do, we do the electrophoresis. What is electrophoresis? सबको पता है ना? Okay, so then we read the sequences. From where we read the sequences? From bottom to top. So what is the principle of electrophoresis? How the molecules move? Based on okay, so these are the chemical modifications. These are reagents used. For example, if I say if I am using dimethyl sulfate, so it will go and cut the G position, G. Okay, and then if DMS followed by formic acid, it will be specific for G plus A. So here you will be getting band in G as well as A link. Okay, so similarly here also for pyrimidine we have hydrazine. Okay, and this is the after loading your reaction, so you will be getting bands like this. So how you will read the sequencing gel? Can anybody read this? What will be the sequence from bottom to top? G, G, A. Can you write on your notebook? आपको प्लीज कुछ समझ में नहीं आएगा तो you can stop me at any point, okay? Because until and unless you will not get the Sanger and the Weizenberg, you will not understand the NGS. Okay, पहले आप basics एक बार आपको समझ में आना चाहिए what is the principle. Okay. So if you see here, if you are getting bands in both G and G plus A, it means G will be there. Okay, and this only if you are getting bands in G plus A. Okay, so here it will be A. So what will be here? C, and this one A, G, not C, T. Here G, C, so this will be your sequence. Okay, so that's how we read the sequencing. Okay, you can cross check it. The principle of maximum Gilbert is very simple. You have to just use some specific chemicals and you can perform the sequencing. But the problem, or you can say disadvantage, is use of chemicals which are not, hazardous chemicals. Hydrazine, which is also known as neurotoxins. So then next sequencing techniques is Sanger, and this is also known as chain termination method. Why this is called chain termination? Because here normally what happens, during polymerizations, addition of nucleotides, okay, releases pyrophosphate and phosphodiester bond forms, and then next nucleotide will be added. That's how chain elongation goes on. What happens here in Sanger sequencing, dideoxy NTPs are used. So this is the structure of normal DNTPs and this is the structure of di-DNTPs. So what is the difference? So here 3' OH and here only H. So what happens during polymerization? This 3' OH act as a nucleophile and it attack on the alpha phosphate. Okay, here PPI is released and this form the phosphodiester bond. Okay, when you will add normal DNTP because of the presence of OH. But if OH is not there, if you are adding DDNTPs, what will happen? No phosphodiester bond will form and then chain will be stopped or terminated. That's why this is called chain termination method. Okay. So here also we prepare 4 set of reaction. I am not going into a very deep thing. So in one tube, for example, you are adding DDATPs, DDCTP, DDTP and DDGTP. Other than this, the common things are you are adding primer, DNTPs, polymerases. It will be in all the tubes. Okay. And here also you are getting different bands. I will show you in this picture. For example, this is the template which you want to sequence. Okay. ये आपका template है. And this, when you are added DDTTP in the medium, what will happen? It will
terminate the reaction wherever that particular nucleotide will happen.
Yes.
For example, it is G. Then it will terminate the reaction at G.
Very good. So the corresponding nucleotide is A. So it will be added here. So one, there may be chance that this it will be stopped here or it will be stopped here. Okay. A के पास तो ऐड करेगा because and after, because your DDTTP have is lacking OH. So after that no DLTPs will be added. Okay, likewise here in DDATP, so this adenine it will be added against T. So wherever T will be there, so you can expect these many fragments. Okay, so similarly for DDTTP, DDCTP, we get different fragments, and then we'll do the electrophoresis. Okay, and then we'll read. But here the difference is the sequence which you are reading in the gel from bottom to top is not your template sequence which you want to sequence. Okay, but this is the opposite strand. So this is about Sanger. So Sanger sequencing is good if you are having small projects. Okay, if you are having some PCR products or amplicon sequencing, it is good. But if you are planning for large genome project, then you have to move for the next generation sequencing platform. So coming to the NGS. So this is a non-Sanger based sequencing technology, and the advantage of NGS over Sanger, if we talk about the speed, so they are significantly quicker. Okay, quicker in the sense in Sanger there is chemical reaction and signal detection. These both are two different processes. But in NGS, most of the versions, these processes are combined. Okay, in Sanger sequencing, you can get maximum read up to 1 KB, but here you can get data I mean more than 300 GB or something like that. So it is also cost effective. Sample size requirement, you need a picogram or nanogram sample. Okay, and then accuracy, because this is very faster technology, and you can have more repeats. Okay, more repeats means more coverage, and it will automatically increase the accuracy. Okay, अभी तक मुझे बताओ क्या समझ में नहीं आया, because I can read the faces, many people not getting what I'm sequencing क्या है, समझ में आया? थोड़ी देर के लिए आप कैमरा ऑफ करो, फिर मैं तुमको थोड़ा sequencing समझ में आ गया सबको? Because this is the training is genomics of agriculturally important pathogens. Okay, so you have to understand the basics. Okay. So what is sequencing? Can you you get it? Sequencing क्या है? कैसे करते हैं? How do you perform sequencing? कोई अरेंजमेंट कुछ नहीं है. एक आपके पास DNA का स्ट्रेच है, ओके, लॉन्ग स्ट्रेच. You don't know the arrangement of nucleotide, how these ATGC are arranged, simply. Okay, so what you will do? There are different methods. In Maxim Gilbert, they are telling that I have some specific chemicals which will cut at G. At least you will be knowing, no, कि इतना बड़ा fragment भी यहाँ